you'll be excited to hear that this is the last Camtasia for the semester. I'm going to go on and talk about gene expression, the process of DNA copying itself in the nucleus through a process called transcription to make RNA, and then the RNA leaving the nucleus going out into the cytoplasm and at the ribosomes forming proteins, and that process is called translation. So transcription first, translation second. Again, transcription is DNA to RNA, translation is RNA to protein. So again, some issues to think about. Proteins are made in the cytoplasm, DNA is in the nucleus, the RNA serves as an intermediate. When we start taking a look at DNA and RNA, there's some major differences. Number one difference is DNA is double-stranded, RNA is single-stranded. DNA uses deoxyribose, RNA uses ribose. The bases in DNA are A, T, G, and C. The bases in RNA are A, U, G, and C. If you take a look at thymine and uracil, you'll see that they're very similar, and so the thymine is replaced by uracil in RNA. The function of DNA is it's the site of the genes. The function of the RNA depends on what the RNA is doing. So some of the RNA that's copied actually carries the code for making a particular protein. There's some, and we call that messenger RNA. There's some RNA that's actually a component of the ribosome, and that's ribosomal RNA. And then there's a third type of RNA, a transfer RNA, that carries amino acids to the ribosome. It actually does the job of decoding your RNA into proteins. So let's start with transcription. Transcription is the making of RNA. It occurs in the nucleus, and I asked the question why. I want you to think about it has to occur in the nucleus because we're starting with DNA, and DNA doesn't leave the nucleus. So what we're going to do is produce a strand of RNA that's complementary to one of the two strands of DNA, and it's done with the help of an enzyme. This enzyme here, RNA polymerase, is the enzyme that's required in order for transcription to occur. So again, DNA is a gigantic molecule. You need to realize that in order for transcription to occur, the RNA polymerase can't just start anywhere along the DNA. It has to actually recognize a sequence that needs to be transcribed. And so the DNA actually has regions that have particular codes called promoter codes, and RNA will bind to DNA at sites where the promoter codes are found. So what happens is when there's a promoter present, RNA binds. When there's a promoter present, RNA polymerase binds. RNA polymerase will unwind the DNA there and begin base pairing our ribonucleotides to the complementary DNA, except it's going to replace uracil with thymine. It's going to synthesize in a 5' to 3' direction, and it's going to continue to synthesize till there's another code that's a termination signal. So here again is a picture of this. RNA polymerase recognizes a promoter, binds to the promoter, unwinds DNA, and builds the RNA, builds a growing transcript of RNA until it reaches a termination signal. At that point, the termination, the RNA polymerase stops binding to the DNA, and the new RNA strand is released. So again, we can separate that out into initiation, elongation, and termination. Again, as soon as the RNA polymerase reaches that termination signal, what happens is the DNA is just going to wind right back up, and the RNA polymerase will go on and transcribe at another location. Okay, after RNA is made, it's released in eukaryotes. The RNA that's made in eukaryotes often contains information that isn't used. Okay, so when we look at the sections of RNA that are transcribed, we sometimes divide them into exons and introns. The introns are sort of intervening sequences that aren't part of the code for making a protein. So one of the things that can happen during the process of post-transcriptional modification within the nucleus before the RNA leaves the cytoplasm is the introns will be removed. We also add caps and tails onto the RNA transcript that help stabilize and allow that RNA transcript to function for a longer period. Okay, again, we talked about RNA playing 3 different roles. So we just made the RNA. What's it going to do? It can carry a message. It can be part of the ribosome, and again, it can help transfer an amino acid, the code from a messenger RNA into the code of an amino acid. These little structures here are meant to exemplify or to show you what a tRNA might look like. So you can see this tRNA is actually reading the mRNA, it's base pairing with the mRNA, and it's carrying an individual amino acid and bringing an appropriate amino acid into place. So again, translation is not going to happen in the nucleus. Our messenger RNA, our RNA, has left the nucleus. We're going to go out to the ribosomes. We can divide it into 3 steps: initiation, elongation, and termination. And we can start with initiation. Initiation always starts with the small unit of the ribosome recognizing a start codon. Now, for your purposes, I don't usually have you memorize too many things, but note that the start codon here is AUG. AUG is always the start codon. AUG tells the ribosome, it tells the transfer RNA when it's time to start making a protein. So AUG is the start codon, and the small ribosomal unit bonds, and then an initiator tRNA will bond with complementary base pairing, and it always brings in methionine. Okay, the large ribosomal subunit will join. Okay, and you're ready to begin the process of translation. So again, what happens after that is going to be elongation. So again, here's our initiator tRNA. It's in one of the sites, 2 sites. There's an A and P site within the ribosome. What's going to happen is another transfer RNA will come in that can base pair. Okay, it's going to recognize a 3-base sequence on the messenger RNA. We call that 3-base sequence a codon. The transfer RNA will recognize that 3-base sequence and will complementary base pair to it, and it will bring along an individual amino acid. Okay, the individual amino acid will be brought into place next to the first amino acid, and one of the first things that's going to happen is a peptide bond will form. Okay, when a peptide bond forms, the growing amino acid chain starts growing on the transfer RNA that's now on the A site, so the transfer RNA that's on the P site can leave. Okay, and once the transfer RNA that's on the P site leaves, the one that was on the A site moves over to the P site, and then the next 3-base pair sequence, the next codon, can be read by the next transfer RNA that will bring along an amino acid. So you keep growing and growing and growing the polypeptide chain. So again, just in words, the second messenger RNA codon base pairs with transfer RNA, peptide bonds form, the second messenger RNA moves to the P site, transfer RNA for the next codon can join. Finally, we're going to have termination. Termination occurs when a codon codes for stops, and it just stops the process of translation. Once a stop codon is reached, the newly formed polypeptide chain will be released. The ribosome will separate from the messenger RNA, and the messenger RNA can continue to be decoded by other ribosomes as it goes along. Now, what's kind of neat about that 3-base code, the codons, is that there is a universal genetic code. And the chart that you see on the right actually is a code for all of the amino acids, the ILE, so it stands for isoleucine, LEU, leucine, VAL, valine. Those are all amino acids. Remember, we talked about there's 20 amino acids that make up proteins. This is the code for the protein. So you can just look at the RNA and read the 3-base pair sequence and recognize which amino acid is going to be brought into place. And this code is identical for all eukaryotic cells. There's a few prokaryotes and some mitochondria that use a slightly altered code, but for the most part, this is it. This is the code for life. Okay. Now again, the key thing we want you to understand is we started with DNA. We said DNA codes for all characteristics. Well, now we know what DNA does. DNA codes for proteins. And so proteins actually are what carry out, or they're the enzymes that cause reactions to occur, that cause us to have the characteristics that we do, or they help our reactions to occur. Mutations are changes in DNA. So mutations, if you think about it, is something changing the DNA. If it changes the DNA sequence, it may or may not change the RNA. Okay, and if it changes the RNA sequence, it may or may not change the protein that's formed. So a mutation can be neutral if it makes no change. It can cause a positive change if it makes a protein work better, or it can cause a negative change. Okay, so mutation is just a change in the DNA sequence. Mutations can be caused by mistakes in copying, or they can be caused by chemical or radiations that interfere with the process of DNA replication. Also, viruses can insert large sections of DNA, and viruses are thought to be one of the ways by which DNA sequences expand. We think about mutations as sort of being a bad thing, but if you think about it more, mutations are the raw material for evolution. Without mutations, evolution could never occur. We can describe mutations as being point mutations, where a single nucleotide is substituted, and again, sickle cell and cystic fibrosis are diseases where we've found a single mutation, a single nucleotide changing and causing a disease. Insertion and deletion mutations can cause changes in the way you read a protein, and a missense mutation causes a protein to stop being produced. Here's a really neat picture talking just briefly, and in this case this is a bacteriophage and a bacterial genome, but it talks a little bit about how a virus can actually have its DNA enter the DNA of an organism. So here we have a bacteriophage that's attaching to a cell, and it's injecting its DNA into the cell. And once it injects its DNA into the cell, the phage DNA can circularize, and it can begin to be copied. And when it's copied, it has the code for making more viruses. And so one of the things that can happen is the phage DNA can cause more viruses to be made. In fact, so many viruses can be made that they actually cause the cell to burst and new viruses released. The other thing that can happen is sometimes the DNA of the virus will just become injected within the DNA of the organism, and in that case the DNA from the virus can actually persist within the organism for many generations. Alright, well there's a quick introduction to DNA transcription translation, what I'd like you to do is look through this a couple times, look through some other videos. Again, this is important concepts, but just kind of give you a clue of how the genetic code is used within cells.